This is Dreamtime Science, where tonight's most interesting discoveries become tomorrow's dreams. I've got something particularly engaging to share with you. You know what's fascinating? I've been thinking about how every tiny cell inside us knows exactly when and how to split itself into two. It's like a perfectly choreographed dance happening inside our bodies, quietly keeping us growing and healing without us even noticing. Tonight, I'm excited to share why this simple act of cells dividing is one of the most remarkable secrets of life itself. If you find peace in these nocturnal discoveries, consider becoming a regular visitor to our quiet corner of science. Share in the comments what topic you'd love to dream about next. First, let's create the perfect space for learning. Cocoon yourself in comfort, release any lingering tension, and let your breath become soft and steady. In the hush of night, when the world softens at the edges and the mind drifts towards reverie, there is a subtle pulse beneath all things, a quiet, tireless rhythm that beats within every living being. It is the hidden music of life, a choreography performed in the depths of every creature, from the smallest blade of grass trembling in a midnight breeze to the vast, intricate mind of a dreaming human. This is the dance of life, and at its heart is a miracle so familiar we scarcely pause to consider its wonder. The division of a single cell. From the earliest days of human thought, people have looked at the world around them and sensed cycles, endings that are not ends, beginnings that seem to arise out of nothing, the return of the sun after night, of spring after a hard winter. In ancient myths, stories of rebirth and regeneration fill the spaces between stars and the cracks in the earth. Osiris, dismembered and restored. Persephone, descending and returning, the phoenix, consumed by flames only to rise again from its own ashes. These are tales born of a deep, almost instinctive recognition that life does not remain static, but is ever renewing, ever beginning anew. What these myths echo in grand, sweeping gestures, the cell enacts in silent, tireless miniature. The drama of division, of one becoming two, is an act as old as life itself, a ritual performed billions of times in the span of a single breath. Yet, though it happens in every moment, it is not mere repetition. Each division is a careful negotiation, an intricate performance choreographed by molecules and chance, memory and mutation. It is, perhaps, the oldest dance in the universe, a ballet in which the performers are neither aware of the audience nor of the stage upon which they move. Consider the cell itself, a drop of life adrift in the sea of existence. To our eyes, it is impossibly small, a speck made of even smaller specks, yet within its boundaries lies the power to shape all that we are. For in the cell is written the code of life, the instructions for how to be a fern or a fox or a person, for how to grow, how to breathe, how to sense the warmth of the sun or the brush of a hand. And it is this code, this secret song, that is passed on as the cell divides, a torch handed from one generation to the next. The process begins with a quiet preparation. Deep within the cell, the nucleus stirs, a structure so delicate and complex it is sometimes likened to a library, sometimes to a vault. Here, the DNA, that double-spiraled blueprint, is carefully copied, each letter of its code reproduced with exquisite fidelity. The result is the formation of two identical sets of instructions, each destined for a new daughter cell. This is not a simple duplication, it is an act of trust, of stewardship, for the slightest error can echo down generations, shaping destinies in ways both subtle and profound. As the DNA is copied, the cell grows, gathering the materials it will need for the coming division. Proteins and organelles are assembled, checked and distributed, each according to its purpose. The cell's architecture is rearranged with almost architectural precision, walls and scaffolds shifting to accommodate the coming split. It is as if a craftsman, preparing to build two houses, carefully inventories every brick, every beam, every nail, ensuring that nothing is left behind and nothing is wasted. Then, at last, the moment of division arrives. The cell enters mitosis, the stage so often illustrated in textbooks and animated in documentaries, yet rarely felt for what it truly is, a moment of profound transformation. The nuclear envelope dissolves and the chromosomes, those X-shaped bundles of DNA, align themselves along an invisible axis, guided by the spindle fibers that stretch and contract like the strings of a cosmic harp. At a signal still not entirely understood, the chromosomes are parted, drawn towards opposite poles of the cell. It is a scene of both tension and grace, a pulling apart that is also a coming into being. As the cell pinches inwards, a furrow forms, deepening until the two daughter cells are finally separate, each encased in its own membrane, each carrying the full legacy of the original. Where there was one, now there are two, each alive, each capable of repeating the process, of carrying the dance forward into the future. This is mitosis, the most fundamental act of continuity, the means by which a single fertilized egg becomes the trillions of cells that make up a human body, the means by which a leaf grows, a wound heals, a new generation rises from the old. Yet, for all its ubiquity, cell division is not merely a mechanical process. It is a story written in molecules and time, a story that echoes the deepest hopes and fears of our species. For in the act of division lies the promise of renewal, the hope that we might begin again, that what is lost can be restored. It is no wonder, then, that cultures across the world have woven the theme of regeneration into their myths and rituals, that we return again and again to tales of resurrection, of cycles, of life springing forth where once there was none. In the quiet of the night, when the mind wanders to the edges of possibility, this theme finds new expression in the stories we tell about the future. Science fiction, that great laboratory of the imagination, has long been fascinated by the idea of cloning, of immortality, of defeating the finality of death through the mastery of cell division. In these tales, the cell is not merely a vessel of continuity, but a gateway to transcending the limits of the flesh. Cloning, in its essence, is the deliberate orchestration of the cellular ballet. The first clone, as imagined by science fiction writers, is a perfect copy, a new being, identical in every way to its original, sprung from a single cell, coaxed into division by human hands. The reality, as we have come to learn, is both more subtle and more strange. The famous sheep dolly, the first mammal cloned from an adult cell, was not a mere copy, but a new individual, carrying within her the marks of her genetic origin, but also shaped by the randomness and complexity of development. Each cell that divided to make her was, in a sense, performing an ancient dance on a new stage, guided by human intention, but subject to the unpredictable music of life. The dream of immortality, too, finds its roots in cell division. If a cell can divide without end, if the body can renew itself endlessly, might we not find a way to escape the inevitability of death? Biologists have sought the answer in the peculiar properties of certain cells, stem cells, which can divide and give rise to many different kinds of tissue, and the enigmatic cancer cells, which seem to have fetihes, or gotten how to stop dividing. In the laboratory, lines of human cells have been kept alive for decades, dividing again and again, outliving the people from whom they were taken. Yet even here, the dance is not without risk. For unchecked division is as dangerous as stagnation, leading not to renewal, but to chaos. There is a tension, then, at the heart of cell division, a balance between continuity and change, between the preservation of what is and the possibility of what might be. Each time a cell divides, it carries forward the memory of its parent, yet it also opens itself to mutation, to error, to the slow drift of evolution. It is this balance, fragile and dynamic, that makes life possible, that allows for both stability and innovation. Within the human body, this balance is played out billions of times each day. The skin renews itself, sloughing off old cells and replacing them with new. The lining of the gut regenerates, cells dividing and migrating to replace those lost to the churn of digestion. Even the blood is in constant flux, with stem cells in the bone marrow dividing to form red cells, white cells, platelets, each with their own vital role. Every act of healing, every growth spurt, every adaptation to a new environment is, at its core, a triumph of the cellular ballet, a testimony to the resilience and adaptability of life. And yet, for all the precision and order, there is room in this dance for improvisation, for the unexpected. Sometimes a cell divides imperfectly, a mutation slips through the careful proofreading, and a new trait emerges. Most often these changes are neutral or harmful, but now and then a mutation confers an advantage, a stronger beak, a sharper eye, a more efficient metabolism. Over countless generations, these small changes accumulate, shaping the diversity of life we see around us. The dance of cell division is thus not only the means of continuity, but also the engine of evolution, the source of all novelty and difference. In the quiet spaces between these grand themes, in the laboratory dish, in the growing embryo, in the healing wound, there is a beauty that transcends utility. The sight of chromosomes aligning, the symmetry of dividing cells, the elegance of a process honed by billions of years of trial and error. It is a beauty that scientists, no less than poets, have tried to capture. A beauty that inspires awe, even in the face of all we have learned. Perhaps this is why the dance of cell division continues to fascinate, to inspire stories and dreams, to draw the curious mind ever deeper into the mysteries of life. For in the simple act of one cell becoming two, there is a glimpse of something eternal, a cycle without end, a promise renewed with every heartbeat. As you settle into the quiet of the evening, with the gentle cadence of your own breath echoing the rhythms of the cells within you, it is comforting to remember that you are, in a profound sense, the product of countless such divisions. Each cell in your body is the heir to an unbroken lineage, stretching back to the dawn of life, each carrying the memory of division and renewal. The dance continues, unseen but unceasing, a testament to the resilience and creativity of life. There is much more to this dance than meets the eye, more layers, more mysteries, more stories to be told. For just as the cell divides and multiplies, so too do the ways in which life can grow and adapt, heal and transform. The journey into the heart of the cell has only just begun, and the next step beckons, soft and insistent, like the pulse of a new day waiting to be born. Beneath the shimmering surface of life's tapestry, where cells hum quietly in the darkness, division unfolds as more than a mere doubling. It is an enigma, a process at once familiar and hauntingly mysterious. We may be tempted to think of cell division as a flawless act, a mother cell giving rise to two identical daughters, a perfect symmetry, a mathematical certainty. Yet, if we peer closer, past the textbook diagrams and tidy explanations, we find a world of imperfection, improvisation, and breathtaking complexity. Let us step into this world where the air is thick with possibility, and the boundaries between order and chaos are as thin as a strand of DNA. Our journey tonight will wind through the labyrinthine mechanisms of mitosis and meiosis, those twin engines of growth and inheritance. We will listen for the subtle mistakes, the stutters in the rhythm that remind us of the true nature of biological replication. The narrative often begins with a single word, mitosis. The term evokes images of tidy pairs, chromosomes lined up like disciplined soldiers on a parade ground, dividing with mechanical precision. In this familiar story, a single cell, having grown to the right size and prepared its genetic material, proceeds through a sequence of stages. First, the chromosomes condense, coiling themselves into tight bundles, each a faithful copy of the original. The cell's nuclear envelope dissolves, and a spindle of microtubules forms, stretching from one pole to the other, like the cables of a suspension bridge. Chromosomes attach to these cables, jittering and aligning at the cell's equator. When the time is right, the spindle fibers tug, pulling each pair of sister chromatids apart. The cell elongates, the chromatids drift toward opposite poles, and at last, new membranes form around each set. The cell pinches in two, and the cycle begins again, a mother's legacy passed to her daughters, generation after generation. Yet, for all its apparent order, mitosis is not a mechanical process. It is a ballet, as much art as science, and the dancers are not immune to missteps. Consider the intricate choreography required. Each chromosome, composed of two identical chromatids, must be precisely replicated and separated. The spindle fibers must attach correctly, not too many, not too few, or the chromosomes may be torn or left behind. Even the timing is critical. A moment's hesitation, a single faulty signal, and the whole performance can unravel. Our eyes are drawn to the rare, the anomalous, a missed connection, a lagging chromosome, a spindle fiber that falters. Such errors are not mere curiosities, they are the seeds of transformation and tragedy. A chromosome that fails to separate can leave one daughter cell with too many copies, another with too few. In the context of a developing embryo, such mistakes can be fatal, leading to miscarriage or genetic disorders. In adult tissue, they may be the first steps on the road to cancer. A single cell, its genetic instruction manual scrambled, dividing uncontrollably. But let us not dwell only on disaster. The very rarity of such mistakes is a testament to the cell's ingenuity. To guard against error, cells employ a host of molecular sentinels, checkpoint proteins, surveillance enzymes, and repair mechanisms, all working in concert to ensure fidelity. If DNA is damaged, the cell may pause, repairing the flaw before proceeding. If the spindle is misaligned, division halts until the error is corrected. When mistakes are too great to mend, the cell may even trigger its own death, a noble act of self-sacrifice known as apoptosis. And yet, for all this vigilance, perfection is never guaranteed. Evolution has struck a balance between accuracy and adaptability, for in the grand scheme of life, a certain measure of imperfection is not only tolerated but required. Variability, after all, is the raw material of evolution. Some errors in division provide the genetic diversity that allows populations to adapt and thrive. The tapestry is all the richer for its occasional snags and knots. Now, let us turn our gaze from mitosis to its more mysterious sibling, meiosis. If mitosis is the steady hand of continuity, meiosis is the croupier of chance, shuffling the genetic deck with each generation. Where mitosis creates two identical cells, each with a full set of chromosomes, meiosis produces 4 unique cells, each with only half the original complement. These are the gametes, the sperm and eggs, destined to unite and give rise to new life. The process unfolds in 2 acts, like the twin movements of a symphony. In the first division, homologous chromosomes, one from each parent, pair up, embracing in a phenomenon called synapsis. Here, the true magic of meiosis is revealed, crossing over. Chromatids from each parent intertwine, exchanging segments in a dazzling display of genetic recombination. It is as if 2 stories, each a record of ancient journeys, are spliced together, creating new tales, new possibilities. This crossing over is not a haphazard affair. Enzymes carefully break and rejoin the DNA, ensuring that each chromosome carries a patchwork of maternal and paternal genes. The resulting diversity is staggering. In humans, with 23 pairs of chromosomes, the possible combinations of maternal and paternal chromosomes alone number in the millions. Add to this the genetic shuffling of crossing over, and the potential for variation is nearly limitless. After the first division, the cell divides again, this time separating the sister chromatids. The result is 4 gametes, each genetically distinct. When 2 gametes fuse at fertilization, the genetic lottery is drawn anew. No 2 siblings, save identical twins, will ever share exactly the same genetic makeup. It is this wellspring of diversity that fuels evolution, that allows species to adapt to changing environments, to survive catastrophe, to thrive in the face of adversity. Yet, as with mitosis, the beauty of meiosis is shadowed by the spectre of error. The most common misstep is non-disjunction, when chromosomes fail to separate properly. The consequences can be profound. An egg or sperm may receive an extra chromosome or lack one altogether. In humans, such errors can lead to conditions like Down syndrome, where a child is born with 3 copies of chromosome 21. Other trisomies, or monosomies, are often incompatible with life, ending in silent miscarriage. The question arises, why, after billions of years of evolution, should such critical processes remain so vulnerable to error? The answer lies in the delicate trade-offs that shape all living things. High fidelity in division demands time, energy, and complexity. Too slow, and the organism may falter in the race for survival. Too fast, and mistakes multiply. The machinery of division is an exquisite compromise, reliable but not infallible. As we contemplate these cellular ballets, another question surfaces, floating like a mote in a shaft of light. How do we know what we know? The machinery of division is so small, so fleeting, that for much of human history it lay hidden from view. Only with the advent of powerful microscopes, of fluorescent dyes and high-speed cameras have we been able to watch the drama unfold in real time. We have peered into the heart of living cells, seen the chromosomes dance and divide, and marveled at the elegance of it all. Yet even now, much remains obscure. The exact molecular choreography, the signals that dictate timing and orientation, are the subject of intense research. Each discovery opens new vistas, new mysteries. Consider for a moment the spindle apparatus, the structure that orchestrates the separation of chromosomes. Built from microtubules, dynamic filaments that grow and shrink in response to chemical signals, the spindle is assembled anew every time a cell divides. How does a cell know when and where to build this structure? How do the microtubules know which chromosome to attach to, and how do they generate the force required to pull chromatids apart? The answers are encoded in cascades of proteins, in feedback loops and molecular motors, in the restless motion of atoms and molecules. We glimpse the outlines of these mechanisms, but the full picture eludes us, shimmering tantalizingly just beyond reach. And what of the repair systems, the cellular guardians that patrol the genome? Here too we find complexity layered upon complexity. DNA repair is not a single process, but a suite of pathways, each tailored to a specific kind of damage. There is nucleotide excision repair, which snips out UV-induced lesions, mismatch repair, which catches errors made during replication, homologous recombination, which uses a spare copy as a template to fix breaks. Each pathway is policed by an army of proteins, each with its own quirks and vulnerabilities. Sometimes the repair mechanisms themselves go awry, introducing new errors even as they try to fix the old. In certain cases, the cell's response to damage is not repair, but surrender. When the genetic code is too badly broken, a cascade of signals triggers apoptosis, programmed cell death. The cell shrinks, its DNA chopped into fragments, its contents packaged for removal by scavenger cells. This self-sacrifice protects the organism as a whole, preventing the proliferation of damaged cells that might spawn tumors or disrupt development. But not all cells are so obedient. Some escape the controls, refusing to die when called upon. These rogue cells may acquire further mutations, dividing recklessly, invading tissues, hijacking blood vessels. This is the tragic story of cancer, a tale written in the language of broken cell division. Amidst this drama, there are moments of quiet wonder. In the earliest stages of human development, a single fertilized egg divides and divides again, launching a cascade of mitotic events. The cells proliferate, migrate, specialize, giving rise to the tissues and organs of the body. Each division is a leap of faith, a roll of the genetic dice, a negotiation between order and randomness. And through it all, the genome is both preserved and transformed, the past carried forward even as new possibilities emerge. There is, too, the mystery of aging. With each division, the ends of our chromosomes, the telomeres, grow shorter, a slow erosion that limits the number of times most cells can divide. This built-in clock may help protect us from cancer by preventing cells from proliferating endlessly, but it also means that our tissues gradually lose the ability to renew themselves, a subtle unraveling that underlies the process of aging. Some cells, stem cells, germ cells, possess the ability to maintain their telomeres, to divide almost without limit. In these cells, the boundaries between youth and age are blurred, the promise of renewal always present. The mechanisms by which they achieve this are still being unraveled, another reminder that the story of cell division is far from complete. As our gaze lingers in this half-lit realm, it becomes clear that the enigma of division is not just a scientific puzzle, but a reflection of life itself, fragile, resilient, prone to error, yet endlessly creative. The cell's dance is never entirely predictable. The steps may falter, the rhythm may change, but the music continues, echoing through time. In the quiet spaces between divisions, as chromosomes uncoil and the cell prepares for its next act, there is a sense of anticipation. What will the next division bring? What new errors? What new possibilities? The story is unfinished, the questions still whispering in the darkness, inviting us to look closer, to listen more carefully, to wonder at the dance that sustains us all. And in these mysteries, the stage is set for the next layer of the tale, where division is not merely a process, but a force that shapes the very destiny of life. The yearning to peer into the mysteries of life is as old as curiosity itself. For centuries, the drama of cell division unfolded unseen, a secret ballet behind the veil of flesh and leaf, invisible to the unaided eye. Yet, the story of how humanity has gradually decoded this dance is one of patience, ingenuity, and countless tiny revelations. Tonight, let us wander through the illuminated halls of inquiry, where each tool, born from the marriage of imagination and necessity, has cracked open a new window into the living world. The journey begins in the 17th century with a simple apparatus of glass and brass, the microscope. Imagine, if you will, the hand of Antonia van Leeuwenhoek, steady and unhurried, guiding a drop of pond water beneath his handcrafted lens. The world revealed to him was one of astonishing vitality. Tiny animalcules, darting, gliding, multiplying. With each twist of the focusing screw, he glimpsed a universe previously hidden, and in his notebooks he described what could only be the earliest observed hints of cellular division, though its full meaning would not bloom for many decades. The microscope, in its infancy, was a modest device, limited in power and clarity. But as glassmakers honed their craft and scientists pressed for sharper images, the instrument grew in sophistication. By the 19th century, the compound microscope, layering lenses for greater magnification, allowed figures like Matthias Schleiden and Theodor Schwann to see, in plant and animal tissues alike, the repeating motif of the cell. The cell theory was born, a bold declaration that all life is composed of these fundamental units. Still, the dance of division remained elusive. Cells could be seen, yes, but their inner workings were shrouded by the fog of poor resolution. The process by which one cell became two was like the fluttering of wings concealed behind frosted glass, mysterious, suggestive, incomplete. It was not until the late 19th century that the curtain lifted further. Enter Walter Fleming, a German anatomist with a patient eye and a penchant for dyes. With aniline stains, he coaxed the chromosomes, the bearers of hereditary information, into view, their dark threads twisting and parting within the nucleus. Through his microscope, Fleming chronicled the stages of what he aptly named mitosis, from the condensation of chromosomes to their elegant separation and the birth of twin nuclei. His meticulous drawings, rendered in ink and graphite, are as much works of art as they are records of scientific discovery. In his hands, the microscope became both a portal and a paintbrush, capturing the choreography of division for the first time. But how to reach beyond what is merely visible? How to touch the mechanisms beneath the surface, to tease apart cause and effect? Here, the ingenuity of experiment comes to the fore. In the early 20th century, researchers began to realize that watching was not enough. They devised ways to intervene, to nudge and perturb the cell and observe its response. Hans Spemann, with a steady hand and a strand of baby hair, performed delicate ligations on salamander embryos, separating single cells and observing their fates. His work revealed the remarkable plasticity of early embryonic cells and hinted at the chemical signals that orchestrate division and differentiation. The micromanipulator was born, a tool that allowed the scientist's touch to reach into the very heart of living matter. At the same time, advances in staining and fixation techniques granted ever sharper glimpses into the cell's architecture. Camillo Golgi's silver chromate method, for example, illuminated the complex web of the Golgi apparatus, while Jan Perkinya's observations of protoplasm, what we would now call the cytoplasm, gave substance to the cell's interior. The cell, once a shadowy outline, was now a world of structures and compartments, each with its own role in the drama of division. The 20th century saw the birth of new forms of light. The advent of fluorescence microscopy transformed the act of observation, allowing scientists to tag specific proteins with glowing markers and watch their journeys through the cell. Fluorescent dyes, and later genetically encoded fluorescent proteins, like the famous green fluorescent protein, GFP, became lanterns in the cellular night. Under the fluorescence microscope, chromosomes gleamed in bright colors, the spindle fibers shone like silken threads, and the movement of molecular actors could be traced in real time. These glowing images were not only beautiful, they were revelatory. For the first time, researchers could see where proteins congregated during mitosis, how they assembled the machinery that pulls chromosomes apart. They could watch the nuclear envelope dissolve and reform, the cytoskeleton bend and reshape itself, the membrane pinch and cleave. Each experiment was an act of revelation, a peeling back of the layers of mystery. Yet even the finest light microscopes are bound by the laws of optics. The wavelength of visible light sets a hard limit on how closely 2 points can be distinguished, a barrier known as the diffraction limit. For generations, cellular structures smaller than about 200 nanometers remained blurred, their details lost in the haze. Then the electron microscope arrived, wielding beams of electrons instead of photons. The transmission electron microscope, TM, with its sub-nanometer resolution, transformed cells into landscapes of unimaginable detail. Mitochondria, ribosomes, the double helix of DNA. These were no longer abstractions, but intricate architectures rendered in shades of grey. The scanning electron microscope, SEM, added depth, sweeping over cell surfaces and revealing their contours in breathtaking topography. With these tools, scientists trace the steps of mitosis and meiosis with ever greater precision. They saw the microtubules of the mitotic spindle latch onto chromosomes, the kinetochores act as molecular winches, the contractile ring squeeze the cell in two. The choreography of division, once a distant rumour, was now a spectacle laid bare, but observation alone cannot always reveal function. To truly understand the machinery of cell division, researchers needed to break it, to remove or mutate individual components and watch the consequences. The dawn of molecular genetics brought this power to their hands. In the mid-20th century, the fruit fly Drosophila melanogaster became a favorite subject. With its rapid generation time and easily manipulated genome, the fly allowed scientists to induce mutations and study their effects on development. Through painstaking work, they identified genes that controlled the cell cycle, the progression of stages from one division to the next. In yeast, another model organism, researchers isolated temperature-sensitive mutants, cells that could divide normally at one temperature but faltered at another. By shifting the environment and observing the resulting arrests, they mapped out the molecular checkpoints that guard the passage from phase to phase. The language of the cell cycle emerged: cyclins, cyclin-dependent kinases, checkpoints, and signals. Through a mix of genetic trickery and biochemical analysis, the cast of molecular players came into focus. The Nobel Prize-winning work of Leland Hartwell, Paul Nurse, and Tim Hunt in the 1990s illuminated the master regulators, the proteins whose rise and fall orchestrate the entire affair. Without the ability to manipulate genomes, to create, delete, or alter genes at will, these discoveries would have remained out of reach. Meanwhile, the tools for peering into the cell grew ever sharper. Confocal microscopy, with its ability to focus laser light at precise depths, allowed the construction of three-dimensional images of dividing cells. Super-resolution techniques like STED and palm microscopy shattered the diffraction limit, offering views of structures once thought forever invisible. Now the proteins at the kinetochores, the dynamics of spindle assembly, the movements of individual chromosomes could be tracked with astonishing fidelity. And yet the mysteries did not diminish. Each new tool revealed complexities undreamt of. The cell was no simple automaton, but a responsive, adaptive system, its dance choreographed by 1,000 interlocking feedback loops. To understand not just the what, but the how, scientists turned to the power of chemistry. Through the use of small molecules, drugs that interfere with specific steps of division, they could pause the process at will, capturing cells in metaphase or anaphase, freezing the dance mid-step. Colchicine, for example, binds to tubulin and prevents microtubule formation, halting mitosis and enabling the study of chromosome behavior. Taxol, originally isolated from the yew tree, stabilizes microtubules and disrupts their normal dynamics, a property that would later make it a powerful chemotherapeutic agent. Chemical biology, with its arsenal of probes and inhibitors, provided yet another window into the living cell. Researchers could pulse cells with labeled nucleotides to track DNA replication, or use click chemistry to tag newly synthesized proteins. The ability to manipulate the cellular environment, to starve cells of nutrients, to flood them with growth factors, to expose them to toxins, allowed the exploration of how division responds to stress and signal. Still, the most profound revolution was yet to come. At the dawn of the 21st century, the twin marvels of genomic sequencing and live cell imaging began to reshape biology once more. The sequencing of entire genomes with dizzying speed and accuracy made it possible to catalogue every gene involved in cell division in organism after organism. Comparative genomics revealed conserved pathways, ancient and unchanging, as well as novel wrinkles on the old theme. The CRISPR-Cas9 system, a molecular scalpel borrowed from bacterial defense, unleashed a new era of precision editing. Genes could be snipped, swapped, or rewritten with ease, allowing the systematic dissection of division's machinery. Simultaneously, advances in microscopy made it possible to watch the dance of division unfold in living organisms, not just in isolated cells. Zebrafish embryos, transparent and rapidly developing, became living canvases for the drama of development. Fruit fly larvae, nematode worms, even cultured human tissues, all could be imaged in real time, their dividing cells glowing with fluorescent markers. The boundaries between observation and intervention blurred. Optogenetics, for example, allowed scientists to control protein activity with pulses of light, orchestrating the timing of division at will. And as data accumulated, images, gene sequences, molecular interactions, computational tools emerged to make sense of it all. Image analysis algorithms tracked the trajectories of chromosomes and spindles, while mathematical models simulated the push and pull of forces within the cell. Artificial intelligence, trained on terabytes of data, began to uncover patterns too subtle for the human eye, suggesting new hypotheses and guiding fresh experiments. Through it all, the spirit of the enterprise remained the same, a mixture of awe, patience, and relentless curiosity. Each tool, from the simple magnifying lens to the genome sequencer and the super-resolution microscope, has extended our senses and sharpened our questions. Each experiment, whether a crude manipulation or an exquisitely targeted intervention, has chipped away at the wall of ignorance that once separated us from the heart of the cell. Yet for all our cleverness, the dance of division is not a solved puzzle, but a living, evolving art. New technologies beckon from the horizon: single-molecule tracking, quantum imaging, nanoscale probes that may one day record the flitting of ions and the folding of proteins in the midst of mitosis. The cell remains both familiar and strange, a universe in miniature whose secrets yield only to persistence and imagination. And so, as the night deepens and the world outside grows quiet, let your mind rest on the image of the scientist peering through the lens, steady and intent, tracing the pattern of chromosomes as they part and rejoin. Let the glow of fluorescent proteins and the hum of sequencers fade into the background, and imagine what wonders might yet be glimpsed with the tools not yet invented, the experiments not yet dreamed. Beyond the reach of today's glass and silicon, new frontiers shimmer. How do cells sense their own geometry or measure the passage of time? What unseen signals guide the synchrony of tissue, the harmony of organs, the healing of wounds? In the darkness between discoveries, the next questions await, silent, patient, and full of promise. In the quiet hours, when the world hushes and the mind floats between waking and dreaming, one might sense, beneath the ordinary pulse of existence, a more ancient rhythm. It is the hum of life's perpetual renewal, the ceaseless choreography of cell division. The universe itself seems to breathe through this process, old as the primordial seas, yet updated and renewed with every living moment. Here, at the reflective edge of science and philosophy, we linger and gaze inward, asking not just how cells divide, but what it means that they do. Each of us is, in a sense, the sum of countless acts of division. We began as a single cell, a fertilized egg, swollen with the promise of the future. That cell, by dividing, set the first domino toppling, each split giving rise to more complexity, more intricacy, more self. Our hands, tracing the lines of a beloved poem or the contours of a loved one's face, are the product of innumerable cellular doublings, each a silent echo of that first division. We carry within us a lineage of cells that have survived, multiplied, and specialized, sometimes adapting, sometimes failing, always striving toward the next dawn. Pause here and consider, why does life multiply at all? Why not remain a single, undivided whole? The answer is both practical and profound. Division allows for growth, for repair, for the possibility of diversity. It is the means by which a lone cell becomes a symphony of tissues, a forest of neurons, a beating heart. Through division, life escapes the confines of monotony, branching into forms unimagined, each new cell carrying the blueprint of its origin, yet open to the improvisations of chance and necessity. Yet this multiplication is not without its paradoxes. In the heart of the process lies a tension between immortality and mortality, continuity and fragility. The story of cell division is the story of inheritance, but also of loss. Take a moment to contemplate the immortal thread woven through our cells. Some lineages have persisted unbroken for billions of years. Each time a cell divides, it passes on its DNA, a molecular text copied with astonishing fidelity. In a sense, this is immortality, the information, the instructions for building life, hopping from cell to cell, generation to generation. The germline cells, those destined to become eggs and sperm, are the custodians of this thread, linking ancestors to descendants in an unbroken biological chain. Through them, life persists. But for most cells, the story is different. Somatic cells, those that make up our skin, our muscles, our organs, are not meant to last forever. Their division is finite, bounded by mechanisms that ensure eventual senescence. Each time a somatic cell divides, its telomeres, the protective caps at the ends of chromosomes, grow shorter, like the burning wick of a candle. When these telomeres dwindle to nothing, the cell can no longer safely divide. It becomes old, weary, and finally ceases to replicate. This is mortality, written in the language of molecules. The philosopher might ask, why would nature build in such limits? Is not the dream of endless renewal a worthy one? Yet here, biology reveals its subtle wisdom. If cells could divide without limit, the body would risk chaos. Cancer, after all, is the result of runaway division, cells refusing to heed the signals that keep the organism whole. The boundaries of division are safeguards, threads that hold the fabric of life together, even as they doom each cell to eventual obsolescence. In this, we find a mirror for our own human experience. We too strive for continuity, for the immortality of ideas, of culture, of memory. Yet our lives, like our cells, are bounded, shaped by the inevitability of aging and death. The cellular dance is both a promise and a warning. Life must balance renewal with restraint, growth with order, creation with dissolution. Consider the process of aging, that slow and inexorable transformation. At the cellular level, aging is a tapestry woven from many threads, the gradual accumulation of mutations, the shortening of telomeres, the build-up of molecular damage. With each division, the risk of error grows, the machinery of repair becomes a little less efficient, tissues lose their suppleness, organs their vigor. The body, once a riot of growth, settles into a quieter rhythm, cells retiring from the cycle of renewal. It is not so different from the passage of the seasons or the slow weathering of stone. Yet even as individual cells falter, the larger organism endures, often for decades, sometimes for centuries. Some cells, such as those in the heart or the brain, are granted a kind of quiescent grace, persisting for a lifetime, rarely if ever dividing. Others are in constant flux, skin cells, blood cells, the lining of the gut, all renewing in a ceaseless tide. Our bodies are mosaics of time, old and new, interwoven, mortality and renewal locked in an intimate embrace. The paradox deepens when we turn our gaze to the phenomenon of cancer. Here, the mechanisms of division are subverted, the carefully tuned brakes discarded. A single cell, freed from the usual constraints, multiplies with wild abandon, forming masses that invade and disrupt. In its quest for immortality, The cancerous cell destroys the very organism that sustains it. It is a cautionary tale, a reminder that unchecked division, however seductive, carries within it the seeds of destruction. Yet from the same wellspring of division arise miracles of healing. When we cut ourselves, cells at the wound's edge leap into action, dividing to close the gap, to weave new tissue where old has been lost. The liver, unique among organs, can regenerate astonishing amounts of its mass, a testament to the powers of renewal encoded in every cell. Stem cells, lurking in niches throughout the body, hold the potential to become many different cell types, reservoirs of possibility summoned in times of need. This tension, between growth and order, between immortality and decay, echoes through the story of life at every scale. From the first stirrings of prokaryotes in ancient seas to the intricate development of a human embryo, division is both the engine of change and the guardian of form. The evolutionary success of multicellularity itself hinges on the ability to control division, to coordinate the needs of the many with the fate of the few. But what of the deeper mystery, the emergence of self from multiplicity? Each of us is not one, but many, trillions of cells, each with its own history, its own trajectory, and yet together they give rise to consciousness, to memory, to love and longing. We are, in a sense, the story that our dividing cells tell, the pattern that emerges from their endless dance. This realization can be both humbling and exhilarating. On the one hand, it reminds us of our fragility, our dependence on the proper functioning of microscopic processes invisible to the naked eye. On the other, it situates us within a grand lineage, a living chain that stretches back to the dawn of life and, with luck and care, extends forward into the future. There are those who dream of transcending the limits imposed by cell division, of halting aging, of banishing death. Even now, scientists tinker with telomerase, the enzyme that rebuilds telomeres, or explore the promise of induced pluripotent stem cells, which can rewind the clock of cellular aging. These efforts speak to an ancient longing, the desire to outrun the shadow of mortality. But perhaps there is wisdom too in the acceptance of limits, in recognizing that the cycle of birth, growth, aging and death is not a flaw, but a feature of life's design. For every end there is a beginning. As old cells retire, new ones take their place. The body is constantly remaking itself, a river whose waters are always in motion, never the same from one moment to the next. In this continual renewal there is poetry, a reminder that change is the only constant, that identity is not a fixed point, but a process. Turn then to the broader view. Across the tree of life, cell division has given rise to astonishing diversity. The same fundamental process that knits together the tissues of a human being also gives rise to the branching filaments of fungi, the leafy green of plants, the vibrant forms of coral reefs. Each lineage has adapted the rules of division to its own needs, sometimes favoring speed, sometimes accuracy, sometimes elaborate patterns of development. The evolutionary arms race has sculpted myriad solutions to the challenge of building and maintaining complex bodies. And yet, beneath this diversity, a common thread endures. The machinery of division, DNA, chromosomes, the mitotic spindle, is conserved across vast gulfs of evolutionary time. In the humble yeast cell, one can trace echoes of the same processes at work in human embryos. It is as if all life shares a secret dialect, a code passed from generation to generation, each species adding its own inflection, its own poetry. If you listen closely, you might hear the resonance of this code in your own life. Growth is not only a biological process, but a metaphor for learning, for change, for the journey from innocence to experience. Aging, too, is more than the accumulation of years. It is the deepening of perspective, the softening of certainty, the flowering of wisdom. Even death, so often feared, is a part of the cycle, making room for new forms, new stories, new possibilities. In the end, perhaps the greatest gift of cell division is not immortality, but continuity, the unbroken thread that links past to future, the assurance that life, whatever its form, will persist. Each cell, in dividing, enacts a small act of faith, that the world is worth enduring, that the story is worth continuing. So we drift in the quiet dark, buoyed by this knowledge. Our bodies are constellations of cells, each tracing its own arc through time. The dance of division goes on, silent and steadfast, weaving us into the fabric of existence. Somewhere, even now, a cell is slipping through prophase, its chromosomes aligning for the leap into the unknown. Somewhere, a new beginning stirs. The night deepens. The questions linger, unresolved but luminous. What is the self, if not the sum of countless divisions? What is life, if not the willingness to begin again and again and again? The answers recede like stars at dawn, but the dance continues, eternal, mysterious, and ever-renewing. As our voyage into the quiet dance of cells softly drifts to a close, we watch their gentle rhythm like a comet tail fading into the hush of space. Let this peaceful unfolding of life's smallest miracles lull your thoughts, and when you're ready, return with me for more Dreamtime tales, where the universe whispers its deepest mysteries.